All right. Inoculation and identification, incubation identification, I'm going to briefly go over. So, incubation, what's the goal? Controlled environment, that's all. Often the idea, oh, we need to incubate microorganisms at 37 because they don't grow at room temperature. Bullshit, they do. Okay? If I will leave those at the room temperature till Tuesday, you're going to have wonderful colonies. Will they grow slowly? Yes, but they will grow. Make sense? So we're not only providing optimal conditions, we can add optimal conditions to the list, of course, but the most important factor is that it's a controlled environment. So we can safely reproduce the results. Whether we are reproducing our own results, I want to repeat the same experiment 2 weeks later. Does that make sense? And I don't have to think, oh crap, what was the room temperature 2 weeks ago? Because it's not constant here. But I know 2 weeks ago I grew them at 37. Now I'm going to grow them at 37, right? And if I do my experiment and Jessica wants to do her experiment, she's looking at the manual, okay, he grew it at 37, I'm going to grow at 37, we can directly compare results. She doesn't have to guess what was the room temperature here, right? Make sense? That's an important part. Now, the third I is isolation. There are 3 main techniques of isolation, and I will briefly describe them. It's the streak plate, pour plate, and the spread plate. You're literally going to be doing streak plate next Tuesday, okay, with your results from there. So how does it look? I don't want to leave my little place here. So imagine this is the Petri dish. It is not, but I'm going to use it as an example. So here's what you do. Why do we need to isolate? So imagine you have a colony, okay, and it's yellow. And you're not sure if this colony is truly yellow, if it's a mix of white and orange. Does that make sense? So how can you know? Use the sterile wire loop. You tap the colony, and you make a little streak. Imagine that this is the agar. You make a little streak on the agar. You sterilize the loop in the flame. Take a small smidge of that first streak and spread it over an uninoculated surface on the agar. So you're thinning it out. And then you're thinning it out again. And you're thinning it out again. So basically the whole purpose of these isolation techniques is to separate bacterial cells far enough so you can be sure that your colony contains only one type. Does that make sense? You see what I'm saying? Like, okay, you have a crowd which has a weird color, crowd of football fans. And you start to separating them, and finally you see that, no, that's all Cleveland Browns fans. Or you start separating them, and suddenly you see, oh, there's a mixture of Cleveland Browns and Pittsburgh Steelers fans. Because now when you start separating them, you see like orange versus yellow. See what I'm saying? So same idea in isolation here. Streak plate is go-to technique. Pour plate and spread plate. So what is that? You take a sample of your culture, whether it's a liquid culture, you can take a colony. You put it, in case of pour plate, you put it in the liquid agar. That makes sense? And then you make serial dilutions using the liquid agar. And you pour each dilution into the plate. The farther you dilute, the less cells you have. The more spread there will be in each plate, better isolation you're going to get. Make sense? With a spread plate, you make serial dilutions not by the agar, but by broth. What you do afterwards, you take a sample of dilution, add it to the plate, and then using a fancy device named hockey stick, distribute the solution on the surface. Okay? Same idea, slightly different technique. So again, match the description with the term, right? Now, last little bit, and this is the type of statement that memorize it statement. We're going to continue so inspection. What does it imply? Inspection includes things like shape, color, size of the colony. Okay, so you can characterize it like, you know, small, round with entire margin, convex elevation, blah, 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 you know, glossy appearance. So basically you describe a colony. And then you can take your microorganism and put it under the microscope. So microscopy is also a part of inspection. Usually inspection does not give you the final answer, what it is. So you can characterize it as the gram-positive cocci that produce white, round colonies with entire margin, but it doesn't give you the name of the species. Is that clear? Now, let me explain what you need to know about microscopy. We're not doing staining today, but I want to go over microscopy. This is the part that I don't want to go, it's not something that I can do better job than the table in the lecture notes. So let me explain. There are 7 types of microscopy in the lecture notes. Bright field, dark field, phase contrast, differential interference contrast, fluorescent, confocal, and 2-photon microscopy. What do you need to know? Basic description of each. So for instance, I tell you, specimen on the bright background, bright field. Specimen on the dark background, dark field. Optical trickery that allows you to observe live specimens at the very high contrast. You think, okay, it's either phase contrast or differential interference contrast. I promise I will not have like really confusing answers being together. Like when you have to, what did he think? You're here not to read my mind, you're here to answer questions, right? Couple of things that I want to highlight. So DIC, differential interference contrast, makes fake 3D sorts of live images. Confocal produces almost a 3D image. Here's how. It takes slices of the slices of the specimen, optical slices. You see what I'm saying? So in confocal microscopy, like in CT scan, the microscope takes multiple pictures at different angles, combines them in the software, producing a 3D image of the specimen. Does that make sense? And finally, the 2-photon, what you kind of need to know, because it can be like a lot, allows you to produce high quality 3D images of living microorganisms in the thick, often like tissue specimens, living tissue specimens. Does that make sense? Like, it's the best light microscopy we probably have. So you can visualize microbes in the bloodstream interacting with blood cells in the real time.
What's that?
It's 2-photon microscopy. Costs a fortune. $250,000. More than that, actually. One setup. Does that make sense? Now, when I say there's 7, I mean it. There is no hidden message. I'm not going to ask. This is my principle. I tell you, you need to know this. I reserve the right to ask any of this, but not more than this. Okay? So, for instance, there are many types of electron microscopy. There are only 2 in the notes. Those are 2 that I can ask. Transmission and scanning. Clear? What do you need to know? The difference. Scanning microscopy. What do you use it for? To appreciate the surface and the shape. So imagine that like you look at this mug using scanning microscopy and you say, okay, kind of smooth, there's a little thingy here, I don't know, the sliding closing thing on the lid, there's a handle, I can appreciate that. What's inside? I have no idea. Maybe it's like rock solid inside, maybe it's all filled with, I don't know, molten gold or something, I have no idea, right? So you appreciate the shape. What does transmission microscopy do? It basically slices it optically and says at this particular level, there's coffee. At this particular level, there's no coffee. You see what I'm saying? So if you need to look inside the cell or inside the virus, you use transmission. If you want to look at the surface and the shape, you use scanning. Questions? Good? All right.